As you know, lysosomes were discovered by Christian Didiov in 1955. Okay, lysosomes were first discovered by Christian Didiov in 1955. These are unit membranous structure which enclose a dense stroma and a large vacuole. Okay, unit membranous structure that is made up of single membrane that encloses a dense stroma and a large vacuole. The large vacuole contains hydrolytic enzymes. Remember it, the large vacuole contains hydrolytic enzymes. Now, as far as the function of hydrolytic enzymes are concerned, it is digestive in function. So, what it does? It digests the materials taken up from outside as well as obsolete components of the cell itself. So, digest all the materials taken up from the outside as well as the obsolete components which are not needed by the cell. Got it? Another important thing is that lysosomes show polymorphism, that is they show variation in size and shape due to differences in materials taken up for digestion. Got it? They show polymorphism, they show variety in shape and size. Why? Because they differ in materials taken up for digestion. Okay. So, now let us come to the types of lysosomes. Lysosomes are of four types: primary, secondary, residual bodies and autophagic vacuoles. So, these are the four types: primary lysosomes, secondary lysosomes, residual bodies and autophagic vacuoles. Let us come one by one. Primary lysosomes. Now, primary lysosomes are the newly formed lysosomes that contain inactive enzymes. So, whatever the enzymes are found, they are in inactive stage. Okay, inactive stage. These are small vesicle-like structure formed from Golgi complex. So, primary lysosomes are the newly formed structures which are vesicle-like structures formed from Golgi complex, and they contain enzymes which are in inactive state. Now, come to the secondary lysosomes, which are otherwise known as heterophagosomes, otherwise called as heterophagic vacuole. These are formed when pinosomes or phagosomes combine with primary lysosomes. I will discuss about pinosomes and phagosomes while talking about the membrane transport. Okay. So, these are formed when pinosomes and phagosomes they combine with primary lysosomes. Now, they contain both digestive enzymes and the materials to be digested. So, they contain both digestive enzymes and the materials to be digested. Got it? So, this is what the secondary lysosome, otherwise known as heterophagic vacuole. Okay, contain both digestive enzymes and the materials to be digested. Then let us come to the third one, that is residual bodies. What are residual bodies? Residual bodies are otherwise known as tertiary lysosomes. These are actually nothing but the secondary lysosomes with undigested or indigestible substances. So, after digestion has taken place, the secondary lysosomes with undigested substances is otherwise known as residual body. Okay. The next one is autophagic vacuole. What is autophagic vacuole? Actually, these envelop and attack the intracellular organelles and digest them. Whatever the cell organelles are found inside the cell, the organelles are found, they are being attacked and digested by lysosomes. In that state, we call it as autophagic vacuole. Okay. These are formed by the union of many primary lysosomes around old and dead organelles. Okay. When many primary lysosomes they get associated around old and dead organelles to form autophagic vacuoles. Got it? So, what are the various types of lysosomes? Lysosomes are of four types, not really types, they occur in four morphology. That is the reason why we call it polymorphic in nature. Okay. Primary, secondary, then residual bodies, then last one is your autophagic vacuole. Now, let us come to the functions of lysosomes. So, what are the functions of lysosomes? First one is intracellular digestion. So, lysosome helps in intracellular digestion. How does it help? It actually digests the stored materials like proteins, fats, etc. to provide energy to starving cells. Got it? So, it digests the stored materials like proteins, fats, etc. to provide energy to starving cells. Got it? So in this way it helps in intracellular digestion. Next one is autolysis. Autolysis. What is this autolysis? Actually, lysosome helps in the digestion of intracellular organelles. Okay, the autophagic vacuoles, what I have already discussed. Digestion of intracellular cell organelles by the process of autophagy, okay, which causes death of the cell. Okay, thus lysosomes are otherwise known as suicidal bags. Remember it, lysosomes are called suicidal bags. Why? Because they cause digestion of intracellular organelles by the process of autophagy, which causes the cell death. So they are called as suicidal bags. So first function is intracellular digestion, second one is autolysis, then last one is extracellular digestion. What is this extracellular digestion? In this case, the enzymes of lysosomes are actually liberated outside for digestion of substances. So they are liberated outside. For example, lysosomes of sperm. Let us consider about lysosomes of sperm. It liberates enzymes which dissolve the protective covering or coating of ovum. Got it? The lysosomes of sperm liberate enzymes which dissolve the protective coating of ovum. Got it? Allowing the entry of sperm nucleus into egg. This allows the entry of sperm nucleus into egg so that fertilization might happen. So in this way extracellular digestion occurs through lysosome. Got it? So this is what the function of lysosome is all about. Now let us come to the next one, that is vacuole. You have heard about vacuole. These are actually non-cytoplasmic. They are separated from cytoplasm by a membrane called tonoplast. So now come to vacuoles. Now these are actually non-cytoplasmic. You know about vacuoles. These are non-cytoplasmic. Then they are separated from cytoplasm by membrane called tonoplast. Remember, the tonoplast is the membrane which separates the vacuole from cytoplasm. It's a selectively permeable membrane. Remember it. It comprises 80 to 90% of total volume of the cell. So total volume of the cell, 80 to 90% of total volume of the cell is comprised by vacuole. Okay. The vacuolar sap is acidic in nature. Why? Why it is acidic? Due to constant transfer of protons from cytosol to vacuole. Okay. It is acidic in nature due to constant transfer of protons from cytosol to vacuole through H plus ATPase pump. Okay, found in tonoplast. So the H plus ATPase pump that is found in tonoplast, which is a selectively permeable membrane, helps in the constant transfer of protons from cytosol to vacuole, which makes the vacuolar sap acidic. Got it? Now come to functions of vacuole. It acts as storage organ, storing pigments, salts, toxic compounds, etc. Okay, acts as storage organ. The second one is it stores ions and organic solutes. It stores ions and organic solutes. Hence, endosmosis occurs. You know endosmosis? Okay, I will, even if you do not know, I will let you know about endosmosis. That is osmotic withdrawal of water. Endosmosis means osmotic withdrawal of water. So endosmosis occurs. This results in turgor pressure, resulting cell wall extension and production of large cells. So resulting in cell wall extensions and production of large cells. Got it? Now, as far as your vacuoles are concerned, vacuoles actually help in the vacuoles are of many types. They can be sap vacuole, they can be gas vacuoles or air vacuoles, they can be food vacuoles, they can be contractile vacuole. So we are not going into details of all these things. So as far as vacuoles are concerned, this is the function of vacuole. Okay. So I hope the endomembrane system will be very easier for you to understand, and we will meet again in another video. Thank you.